Welcome, everyone. My name is Michelle Georges. I want to start by thanking you all for joining us today. We at the Carroll Center for the Blind are honored to be partnering with SpliceBio, our sponsor for today's session, to discuss SpliceBio's work in pioneering a novel gene therapy for Stargardt disease. So thank you, SpliceBio, for sponsoring today's event and for partnering with us to bring everyone this very valuable information. Now, before we get going, I do want to take a moment to go over just a few housekeeping items. To start, we are offering ACVREP credits for today's session. You will need to email development@carroll.org after the session with the entrance and exit codes. The entrance code is GREEN, GREEN, green like the color, and I will provide the exit code at the end of the session. As a reminder, you may have heard, the session is currently being recorded and will be shared with everyone after for those who may have missed it or would like to take another listen. Now, we do have the Q&A feature currently enabled on this Zoom webinar, so we do encourage you to pop your questions into that chat box at any point throughout the presentation. And at the end, during the Q&A portion, we will try our very best to get through as many questions as possible, and we will follow up with any questions that we cannot get to. Now, without further ado, I would like to turn it over to our two wonderful speakers who will be helping us understand all about SpliceBio's incredible work and will be helping us throughout today's session. John, I'm going to pass it over to you.
Thanks, Michelle. Appreciate it. And thank you to you, Michelle, and thank you to the Carroll Center for arranging this webinar and for everyone for registering and joining. It's a shame I can't see you all, but I know you're out there. So we're very honored to be with you, of course, and for the opportunity to speak about SpliceBio and speak about what we're doing and what we've been doing personally for myself and Farida for the last couple of years. So if you just move one slide then, Farida, thank you very much. I think we're just going to kick off with some introductions. And, you know, it feels a little bit self-indulgent, I must say, when I do things like this and I see my name up there, but we'll give it a go and bear with us. So my name is John Tobin, and I've got about 30 years now in the industry, which has just flown by, it seems. And I've spanned multiple therapeutic areas over that time, including general anesthesia. I actually spent quite a long time in migraine headache, as well as Parkinson's. And then I started to move into ophthalmology, and I ran global programs in diabetic macular edema and dry AMD. But in the last 9 to 10 years or so, I've been specifically looking into and working in inherited retinal disease, and even more specifically, in most cases, looking at gene therapy and how gene therapy could be a potential treatment for the likes of choroideremia, X-linked RP, LCA10, and Usher's. As you may have noticed from my accent, I'm based in the UK. I actually live in a town called Reading, which is in the county of Berkshire. And if you know your UK geography, it's just west of London and just south of Oxford. So I'm in around about that kind of middle ground area, if you like. My role at SpliceBio is I'm the Vice President of Clinical Operations. And I've been here for about 2 years, and I'm really focusing on the execution of our gene therapy studies for SB007, which is kind of a bit of a bland name, but it's a little code we use to describe our gene therapy, and specifically in the treatment of Stargardt disease. So that's me, and I'll just hand over to my friend and colleague Farida to talk through her bio.
Thank you, John. Hi, everyone. My name is Feride Şahin. I'm a medical doctor, but I'm not an ophthalmologist. I'm a general practitioner. So I have been working in the industry for 26, 27 years now, and I've worked in different therapeutic areas like cardiology, rheumatology, dermatology. But the last 15 years, I have been working in ophthalmology. Under ophthalmology, I worked on different diseases like diabetic macular edema, age-related macular degeneration, glaucoma, dry eye, uveitis, retinitis pigmentosa. I'm based in Geneva, Switzerland, and at SpliceBio, I've been working in SpliceBio for around 13 months now. I work as Senior Director Clinical Development. My major role is I'm the medical monitor of the clinical studies of SB007 in the treatment of Stargardt disease, which John will be speaking about in the following slides. So what does a medical monitor do? I can say I act as a safety guardian for the study, making sure that the participants' health is protected. Of course, throughout the trials, of course, we work with very experienced ophthalmologists, and they provide the care to the participants. But I am like a second eye, checking the health of the patients from the database to ensure that the study, the health of the patients are protected, and the study is ongoing according to the ethical and medical principles. So back to you, John.
Thanks, Farida. Okay, so Farida's based in Switzerland. I'm based in the UK, but SpliceBio is actually based in the beautiful city of Barcelona. And so that's lucky for Farida and I, because it means we get to go there from time to time. And, as you may see from this picture, it's always sunny. There's lots of sports going on. There's lots of beautiful young people. So not 100% sure how I got the job, but regardless, it's I do my best. So Farida, if you just want to just click on the technology that we work with is actually from Princeton University, from Tom Muir's lab. And our CEO, he is from Barcelona. He worked in Princeton for many years. And when he set up SpliceBio, he took it home, basically. So that's why we're in Barcelona. There's around 40 employees, about 45 employees now, I want to say. And earlier this year, after an initial fundraising effort of 50 million in Series B, which is our first round of funding, we have 135 million raised now. And that's good for us. Of course, it means we can move our projects forward. And it also means that, that there's good sort of confidence from investors that this is good technology that we're working on, and something they want to invest in. So if you go to the next slide, Farida. So the technology itself is gene therapy, and there are other gene therapy companies out there. I think what makes SpliceBio unique is that we target diseases caused by mutations in large genes, okay? And it's the large genes piece, that's the bit which is tricky for researchers to deal with, all right? But we deal with it through something called protein transsplicing, all right, which is a biological process. And splicing, of course, is the origins of the name SpliceBio that we particularly take advantage of. And we use that to address the genetic underlying cause of the disease, right? So there's a few words there that kind of don't make sense, probably. And but what the session is today is about is just trying to understand what these things mean and why it's important, particularly when we're addressing large genes. If you just go on, Farida, then obviously Stargardt disease, as we've said, that's our front-running program. And this is Stargardt disease caused by mutations in the ABCA4 gene. So based on what I've just said, it's reasonable to assume ABCA4 gene is a large gene, and of course you'd be right. And we know that there's hundreds actually of mutations that can occur in that gene, and potentially even over 1,000 that are disease causing. Okay, so problematic in terms of the size and the potential for pathology. We do have a pipeline. We have other inherited retinal disease programs that we're reviewing. I can't disclose them at the moment, but it's something for the future. Okay. So we've been working hard, our preclinical team have been working hard over several years now on multiple animal models where we test SB007, our gene therapy, and we generate a nice data set within the preclinical environment. And that gives us good confidence. And if you just click on Farida, it gives us good confidence then to produce this data package that we can deliver to the regulators, okay? And it's the regulators who will then give us the approval to move forward into our first clinical trials. And of course, we've done that. We've done that in the US with the FDA, and in the UK equivalent, which is the MHRA, we've also got approval there as well. So lots of acronyms, but essentially regulatory green light to move forward into treating patients. And of course, that's what we've started to do. we've started at the beginning of this year. We started our first in human study, and in parallel to that, we will be running an observational study. And I'll talk a little bit more in detail about what this observational and also the first in human studies actually look like, and what it may mean to even recruit into them. But before then, I think we just want to have a little bit of a deeper dive into what is gene therapy and the principles around it, and how we deploy that specifically at SpliceBio. And for that, I will hand over once more to Farida.
Thanks again. So I'm sure everybody knows how gene therapy works, but let me try to explain it briefly once again. So think of your DNA as a huge manual of instructions. And in this manual, there are sections which are called genes. Genes are like recipes in a cookbook, and your body uses this information, these instructions to prepare, to make the properly functioning proteins which your body needs to function perfectly. So, and when one of the genes are missing, or if they are not correct, then this causes the body not to function correctly, and ultimately this causes diseases. So what we do in gene therapy, think that the gene is like a letter of instructions. So we want to give this to the patient. It is the patient needs this correct gene to function properly. So what we do is we put this letter, we need to put it into an envelope, right? You cannot send a letter without an envelope. So what is this envelope? We use associated viruses. We call them vectors. So these are harmless viruses. They don't cause disease in the body. So we put our letter, the gene, into the vector, into the envelope. Now our package is ready. But of course, we need a delivery truck, a platform to give it to the body. So in eye diseases, we use injections to do that. So it can be a subretinal injection, an intravitreal injection. And this is one-time delivery. So we give it only one time. And then when the body gets this manual of instructions, the cells read them and use this information to make the properly functioning proteins. And the output is, delaying or halting the progression of the disease. So as I said, gene therapy is one and done. So we give the treatment only once. So there are other treatments like pills or injections that you receive every day or every month. It's different because the aim of the gene therapy is not the focus of the gene therapy is not one single symptom of the disease. We are focusing on the root cause of the disease. And the objective, the ultimate aim is to cure the disease. Even if it is not possible always, we can halt the progression or delay the progression of the disease. So coming to Stargardt disease, coming back to Stargardt disease, which is our lead program that we are working on. ABCA4 gene is a gene that produces ABCA4 protein in the body. ABCA4 protein is like a recycling protein. So when the protein works, it is in the light sensitive cells of the eye, and when it works perfectly, there is no problem. But when there is a problem with the gene, the protein cannot be made properly. The correct protein cannot be produced. And in this case, the waste builds up in the eye, and this causes the cells to be damaged and die by time. And as the number of cells that die or damaged increase, the vision gets worse and worse. So by injecting the ABCA4 gene into the eye, we send the correct manual to the eye, and the retinal cells start using this new information to produce the ABCA4 protein. And new ABCA protein helps eye cells to function better, and this helps improving, protecting, or even improving the vision. So John mentioned already about the ABCA4 gene. He said that it's a large gene. Yes, now we have the ABCA4 gene, which is perfectly functioning. It's the correct gene. We want to give it to the body, to the eye, but we have a problem. Because these AAVs, the vectors, have a cargo capacity. And since ABCA4 gene is very large, it is not possible to put it into one single virus. So in SpliceBio, what we are doing is we are splitting this gene, this large gene, into two pieces, two fragments. And to each fragment, we are attaching small engineered protein pieces, which are called inteins. They are produced by SpliceBio. And we are putting these fragments into two viruses. Of course, this doesn't mean that you will get two separate injections. So they are put into the same solution. Now we have our letter, we have our vectors, two viruses, and we will give it to the body. So what is our delivery track? Here we use the subretinal injection, because subretinal injection gives the treatment directly to the location of the damaged cells. It is the photoreceptors, rod and cones, and retinal pigment epithelium. And that increases the possibility of the potential of the effect. So when the vectors are injected, they directly go to the cell nucleus. There, the DNA is transcribed into RNA, and then the RNA is translated into proteins. So now we have two proteins, and they are still separate from each other, and the inteins that I mentioned are still attached to these proteins. And at this point in the cytoplasm, the magic starts. These inteins have affinity to each other, and they bring two protein pieces, two fragments of the protein together, and the full protein reconstitution takes place. And what happens to these inteins? They detach themselves from the protein, and then they are degraded. So, and you cannot find any fragments of the inteins in the eye. And the full-length protein is the functional protein. It functions perfectly like the original protein of the normal, the people who do not have this disease. Yeah, back to you, John.
I think there's a question, Farida.
Yeah, I cannot see it.
I've got it here. Does the injection cause any damage or risk to the eye, such as atrophy?
In the literature, yes, this is possible. Some atrophies are, they disappear after a while. It's not very common, I would say. and it doesn't have an impact on the visual acuity, the vision. Of course, we are making sure that, we are working with very experienced ophthalmologists, and we are trying to make sure that, there is no damage to the patient, to the eye. I don't know if this answers the question, or John, I don't know if you want to add anything.
No, I think that's accurate. I mean, we also inject away from the macula as well, right? So that sensitive area, that sensitive central area, is not impacted by the injection, as because the fluid moves across over time. Okay, good. Well, thank you for that. Oh, I think there's maybe another question. Oh, thank you. Thanks, Amy. Okay, excellent. Farida, do shout if there's some more questions that comes up. I see another one. Does this gene therapy work also for advanced stages of Stargardt?
In our studies, we are enrolling moderate to advanced patients, and of course, we have some inclusion, exclusion, eligibility criteria, but yes, we expect it to work for the advanced patients as well.
Okay, excellent. Okay, thanks then, Farida. So let's just look at the clinical development plan and walk through that. And again, please keep the questions coming. It would be good to keep this as interactive as possible. So we started with an observational study called Polaris. Polaris is the North Star, okay, and it's a star that's guided travelers for centuries now, and we see it as that this study will guide us through the program, okay. So that's the origins of the name. And as we monitor the patients in Polaris, we will transition them, if you just click on, to our phase 1/2 study, right? That's our first clinical trial, which we call ASTRA, and it's our first in human study. There we go.
I am clicking, but I cannot move it. I don't know why.
Yeah, there we go. You've got it. Okay, perfect. So you can see that Polaris will feed into the ASTRA study. ASTRA coming from ad astra, the Latin, which means reach for the stars. So, a celestial theme building up here. Now, as we run our phase 1/2 studies and we treat patients, we will continue to feed patients into Polaris. And that's important because we also need a pool of patients to go into our phase 3 study. Our phase 3 study being the key study, the pivotal study that will generate the data that we'll use to present to the regulators for approval when the time is right. And as you can see, we don't have a name for that study. So, by all means, in the chat, put in some names. And I know we've got 135 million, but it's all accounted for. And so there's no prizes. But who knows? You could see your name up in the study in clinicaltrials.gov one day. All right. If you go on, Farida, to the next slide. So why do we do an observational study? Why is it important to be part of it? So what's key is that it of course improves our understanding of the disease, and particularly in rare disease where there's just not a lot of information compared to more common diseases that's available. And so we can then learn how to measure the disease and how the disease is likely to progress as well, and how we measure that. And that's important when we think about our study endpoints and our study design. And then also, when subjects are in Polaris, they will be profiled very carefully by ourselves and by the study doctor. And that then allows the study doctor on a patient-by-patient basis to really examine and conclude whether they would be a suitable candidate for gene therapy now or potentially in the future. We've spoken about the regulators a couple of times. Of course, one of our key jobs, our key outputs, is to provide a really quality data set to the regulators, all right, so that they can do their approvals, they can do their reviews in an attempt to get an approval at the end. And for the regulators, they really like to see longitudinal data prior to gene therapy and compare that with after gene therapy as well. So a key element to the regulatory submission. And then finally, for SpliceBio, it provides us with a wealth of information. I've already said that it helps us design studies, But it could also help us refine studies as well. If we're learning more and more about the disease in parallel, it allows us to design our phase 3 accurately. It may even have an impact on how we design other studies in other therapeutic areas in the future. Okay, so it provides this real rich data set that is advantageous in so many ways. And ultimately, what we want to do is give the therapy the best chance it has of being approved. Okay, so that it all adds to that. Okay, so the next slide, please. So who are we looking for in Polaris? Well, we're looking for adults at the moment. We do have adolescents, and we will add adolescents into the study. The adolescent cohort is actually very important, and so they are an important part of our clinical program. At this point in time, we're just focusing on adults. And if you just move on, so those people who participate, of course, it's gene therapy, so we need to understand their genetics. So people with genetically confirmed diagnosis will be included. You've probably heard this so many times before, but it's worth just taking a moment to do a shout for getting genetically tested. Okay, so there are ways of doing that. There are ways of doing that for free in the US, in the United States, and all sponsors like us, all companies like us, will want to understand the genetics of anybody coming into the studies. So getting a head start on that was really a good advantage. Okay. However, if that's not the case in Polaris, we do our genetic testing anyway. So we'll take a saliva and a blood sample to confirm the mutations in the ABCA4 gene. Like Farida says, we're looking at subjects with severe to moderate vision. I think you can argue it's probably moderately good vision, actually. So it's a pretty broad range in terms of vision. And then also, we'll be looking for those with a clinical confirmation of diagnosis of Stargardt's. And if you just click on one more, and we use imaging to help us with that and assess that. And within imaging, we can measure the areas of diseased tissue. We can measure, the areas of atrophy. All right, and that's important in determining our understanding of how the disease progresses. And there, of course, there are other study criteria, but these really are the key ones. And the study doctor will help, walk through these criteria and determine any given person as a potential candidate for Polaris. Okay. So what does taking part actually mean? And again, I've said this before elsewhere, but I always think people volunteer for taking part in these studies. It's an invitation for people to give up their time and their medical history and their data to allow us to progress. And without these volunteers, we've got no chance. So they really are the heroes in the story of what we're trying to do and what all other companies are trying to do as well. So I always want to say a thank you to anybody and everybody who takes part. I'll get to the end of this slide. I think a question's just popped up, and there's a number of questions actually. So get the end of this slide, and then we'll have a look at those. So what does taking part actually mean? If you just click on, and we can go through it. But we need subjects to come in every 4 months, so it's more regular than your normal physician, but hopefully not too frequently to be too much of a burden. And that frequency does drop a little bit actually as the study progresses. And then there'll be standard tests and imaging that you may be used to as well. So for example, visual acuity, which is simply reading letters on a chart, which I think we've all done. We want to understand how Stargardt's affects visual acuity for sure, but also how it affects visual acuity in lower light. Okay, so we'll also do this at normal light, and then you actually get to put on some almost like sunglasses to replicate lower light. And then there's standard imaging as well, and that would consist of fundus photography, where we can look directly at the macular area at the back of the eye, and then also to look at cross sections of the eye using SD-OCT, which is spectral domain optical coherence tomography, which gives us these lovely images of a cross section of the eye, painless images, where we can look at all the layers, see how Stargardt's impacts the layers of the retina, and just the general architecture of the eye and of the retina itself. And then if you just move on to one more, yeah, and this is just slightly unusual, which is why I pick it out. It's called microperimetry. You may have done perimetry in the past. Microperimetry is really getting an understanding of the sensitivity, how sensitive the retina is in that central area, in that macular area. And it involves subjects looking into the machine and then just clicking the button every time they see a light. And you get this nice map, sensitivity map, that is superimposed on an image onto the back of the eye, with the dark areas less sensitive, the bright green areas nice and sensitive. Okay, I'll just stop there, maybe just take some questions. I think the next slide is, if you just go on to the next slide, Frida. So, oh yeah, okay. Okay, so I'm looking at a question here. Someone who's 60 years old has been diagnosed with Stargardt's, problematic gene for this disease. Okay. Yeah, go ahead, Frida.
Unfortunately, gene therapies are specific to the genes, so you cannot inject one gene therapy to, for example, ABCA4 therapy to for another genetic disease. So yeah, it's only specific for ABCA4 related Stargardt disease.
Okay, and then the question about the study locations. So what are the study locations? So the countries that we're currently in are the United States, and we're from East Coast through to West Coast. We're in the United Kingdom, and we'll shortly be, actually, no, sorry, we already are in Germany. Now, what I'll do at the end is I'll show you the NCT numbers for clinicaltrials.gov, for the website clinicaltrials.gov, and show you how to just navigate what terms you need to navigate that website in order to find more detail. And you'll see every single site in the US listed within that website. In terms of considering expanding to other countries, yes, we will expand to other countries, more likely in phase 3. And we tend to go to the countries, of course, where we have established inherited retinal disease experts, because that's where the patients go, okay? And that's where there's higher levels of referrals with Stargardt's into those centers. So it's not fully established yet where they'll be. Now, I have to say, and I've done this before in inherited retinal disease, although Stargardt's is more common than other diseases, it's still rare. And what we'll often do is financially support, that is, movement of patients across state borders in the US and across borders, country borders as well. If patients are willing to come on a frequent basis, and we understand, it impacts with family life and work life, and it's not easy. But if they're willing to do that and they meet criteria, then we're willing to help fund, I should say, all the travel. Okay. Do you know, or have you studied epigenetics play a role in Stargardt's?
We didn't study this. Honestly, I assume epigenetics play a role in Stargardt's, but in the literature, I didn't see too much information about this. But as a company, we didn't study it. Yeah.
Okay. Got a few more questions on a different feed here. So can you give an overview of the animal data, especially NHP, any human data, or is this trial the first? Do you want me to have a go, or do you want to have a go first and I can layer on?
Go ahead, please. Yeah.
Yeah, I mean, I'm not, so we don't have the preclinical team here. From the animal data, especially the NHP data, there is, and of course the NHP, this is the non-human primates. is predominantly looking at toxicity. So there's no evidence of toxicity within the NHPs, and of course this is critically what the regulators look for, and what is going to prevent us moving forward into the clinical trial itself. So we've got a clean bill of health that the regulators supported. The human data, we've only just really started, and so again, there's no safety issues, no emerging safety issues that has prevented us from moving forward. So we continue to move forward. Frida, anything to add on that, or we should go to the next one?
No, I think that's all we can say for animal data. Yeah.
Okay.
The next one.
Go ahead. Yeah.
The next one is, what are the biggest risks? Did you have any NHP and other animal models? Yeah, as John mentioned, indeed, from toxicity perspective, NHP studies were really, we didn't see major toxicity issues. And the doses that we use in the humans are selected very carefully. So yeah, this is what I can say. I don't know if you have anything to add, John.
No, I think that's fine.
So
go on.
How can we enroll for the Polaris study? The next question.
Yeah, so I'll share the slide in a moment with the information. I think the key thing is to contact us directly. Like I say, I think we'll conclude with how to contact us, and we can help to at least begin to understand if anybody could be eligible for the Polaris study. And I think the next one about the trials being in the US only, yeah. I think we've answered that.
The next one is, are there any other gene therapy programs out there? Why do you think this one is better or will succeed? Very good question. Yes, there are other gene therapy programs. Of course, I think we have a very good preclinical data package. So we really, count on it. We are selecting the patients very carefully. And we are working with very experienced ophthalmologists, surgeons. So yeah, combining all these, we really believe that it will succeed. But we will see, of course. John, do you have anything to add?
No. I mean, this is, these are mass experiments, right? And that's why we do the experiments, to answer these questions. So we're at the early stages at the moment, but but I think proof of concept is giving us a lot of confidence.
Yeah, and I think maybe we can add that the SpliceBio team is also very experienced. As John mentioned, our CEO is one of the founders of this platform, the technology, and yeah, it's a very dedicated team.
Yeah. And then I think the final question I can see on this, at least on this, oh no, there's a few more come up. Okay, no, that's fine. We can keep going. So based on experience with someone with around 10% of vision remaining in Stargardt's could be part of the study. I mean, I'll have a go at that one, Frida, if you want, and you can layer on top. I mean, it's difficult to say, right? So like I say, the vision eligibility is quite broad, and it is quite severe. But when we do go down to severe, but when subjects come in, they go through best corrected visual acuity. So the optometrist will work hard with lenses to make sure that people are seeing the best that they could possibly see. So when you go to the optician or maybe the ophthalmologist, and you do maybe, for want of a better term, a cruder a visual acuity test, you may not score as high as you would do in a best corrected visual acuity test. So what I'm saying is that I think the best way to answer that is get in contact, we'll get referred to the closest site, and then they would talk that through with you. And then also, if it goes to a screening visit, then we'll know, because we'll collect the data.
So we have plenty of questions. John, maybe you can take this one as well. How come the SpliceBio website mentions you are recruiting 12 to 65 years old for Polaris when you mentioned only to recruit 18 plus?
Yeah, that's right. So I think at this point in time, okay, we'll be looking for 18 plus. It is true that we actually are recruiting 12 to 65-year-olds, but we're just limiting the number of 12-year-olds. Sorry, 12 to 18-year-olds. We're putting a limitation on that number as it currently stands, because when it comes to actually treating patients, you know, the primary goal in these early studies is safety. And it's important to establish safety, I think, in the older population. Okay, it's once we start to get signals of efficacy, we will then start to introduce the younger population. So it doesn't mean that we're not recruiting those patients. I think just today, at this particular point in time, we just need a little bit of an extra boost in the adult group, and that's where we're focusing our attention. I think soon we will open it again back as an opportunity for adolescents.
Maybe I can take the next one. There are 3 questions from the same person. So can you speak about more about the security and effectiveness of the initial studies? So yeah, John will mentioned about the first in human study. We recently started, I would say, and we don't have too many patients. That's why, if from effectiveness perspective, it's difficult to say something. So because we need to follow the patients for a while. From safety perspective, what I can say, we didn't see serious adverse events related to the product so far. The second part of the question is, if you participate, do both eyes need to be injected, or could it be only one? So at the moment we are injecting only one eye. I hope this answers this question. And the last part is, I have a sister with Stargardt as well, but she's outside the US. Can she participate as well, potentially? John, maybe.
Yeah, I think that's it just feeds back to what I said earlier, is that it's not restricted to the US. We're in UK and Germany as well, but even then, there's still opportunity to travel over borders to get you to the sites.
So
all right.
The next one is, how many people will be in each phase? John, do you want to take it?
Yeah. So in our phase 1/2, we were looking at something like 55 patients plus, okay, up to about 70 patients to put in. So a good amount of patients. Polaris, we don't really put a number on it. You know, it's good just to continue to assess data progression over time. Obviously, the bigger the sample size, the better. And then our future studies, we don't know. And that's why things like Polaris are useful, because it's the data from Polaris and from ASTRA together that will allow us then to work out what the sample size needs to be in order to execute the phase 3. I can have a go at the next one as well. Oh, when do you expect initial data readout? Oh, hang on. I've jumped ahead too.
There are many questions. I don't know. Also, there is a request to finish the presentation before answering the questions, John. Maybe you can move forward with the presentation.
Okay.
Thanks so much. That is very kind. Maybe you can
good.
move forward with the presentation, and then we can continue if we have time. What do you
Excellent.
think?
Yes. Yeah. No, that's good. I mean, actually, there's only a couple of slides left. So we've spoken a lot about the Polaris study. We'll just finish off with the ASTRA study, and a lot of this actually has been covered in the questions. So yeah, we're recruiting, like I said, 50 odd patients plus across US, UK, and Europe. And I think we've already covered the fact that these participants are selected from the Polaris study itself. The initial goal is safety, right? That's always, and of course, safety will be a key endpoint and a key measure for every single study we do. And even once, you know, a drug hits the market, it's monitored continuously throughout its lifespan to assess safety. But at this particular point, we're just focusing on safety, and we do it in a very carefully controlled way. I think one of the questions was around how long does these things take? But when you start, you just want to treat a patient, wait a while, monitor, maybe treat another couple of patients before you even think about going to another dose or a slightly higher dose. And then you do it very carefully, again, treat, wait, and so on. So it's a nice long controlled period to make sure that we're assessing safety. Before then, we start to think about signals of efficacy. Does it work? How does it work? How well does it work? That sort of comes a little bit later. And then, so the study itself, this is still with ASTRA. There's 11 visits over 2 years. Okay, so it's a 2-year study. A lot of the visits are clustered around the surgery itself, and that's purely for safety again. So we want to know shortly after the surgery how well the subject's recovering from the surgery itself. So there's multiple visits immediately after that, and then it smooths out a little bit and is less frequent. And then the tests, the kind of tests that we conduct at each of the visits, pretty much what I showed you before in Polaris. Visual acuity, we take lots of images, and we introduce some questionnaires as well and get some feedback about how your vision is affecting your day-to-day activities. So I think the next slide is the last slide, and we can just leave this last slide up, and people can take a photo of it. We can post it on Carroll Center website, wherever you need it, just to get the information you need. But if you want to learn more and you want to get in contact, you can go to our website. There's a patient inquiries form, okay, which you need to complete. Oh, we'll just leave the yeah, that's it. Let's just leave that. And I think on that inquiries form, there's a little, you can note that where you heard about SpliceBio and the studies, and that's useful for us to know. And that will go to our team. You can email our team on clinicaltrials@splice.bio, and it will go to the same team as those who received the form. So whatever you prefer, right? There's no advantage one way or the other. The same group will still review it. And if you're used to using clinicaltrials.gov and you want to get a bit more detail about the eligibility criteria and the study design for both Polaris and ASTRA, you can either just go in there, but in Stargardt's SpliceBio, and then Polaris and ASTRA will pop up, or I've given you the reference numbers there. You can just put that in the search engine and it will come up with Polaris and ASTRA. Okay. Okay. I see Michelle's popped up. Is everything okay, Michelle? We okay to?
No.
I know we've got seven
Yeah.
minutes.
We still have a few minutes to go, so we could probably take maybe 1 to 2 more questions. But I do also just want to note for everyone who has tuned in, all of this information will be sent via email as soon as the recording is available. Usually we try to do it within a day or two, like 1 to 2 business days, just for our team to get the recording ready for viewing. But we will include all of the information that John just shared about how to get in touch and how to access all this information as well. So just to note as well for the guests. Do we have 1 to 2 more questions, or do we feel as though we've covered most of the information?
Yeah, we can take a few more questions if you want.
Yeah, and Michelle, are we able to answer these questions outside the meeting in email or something if we don't get to them?
Yep, absolutely. So we can definitely do that. We'll take some notes. We actually have someone on our team who's kind of tracking the questions, and we'll try our best to kind of note them all down just in case we didn't get to a question. We can always follow up via email as well and kind of share whatever answers we have.
Okay.
Yeah.
All right. We've only got a few minutes, so I'm just trying to be selective here. I'll go ahead, Farida. Go on.
For example, there is a question about the word improve vision that I used, and our participant is asking if it is possible the dead rods or cones can be restored. Of course, if it is dead, it's not possible to restore. But when I say improve, I'm mentioning about the cells, the rods or cones that are still alive, but not functioning properly. So we can have an impact on these, and by that way, we can improve the visual acuity.
Yeah. Okay.
Well,
Um,
thank you both so much. I think we might have to wrap it up soon. I'm just noticing the time. We have a few more minutes.
okay.
I just wanted to take a moment to thank you both for taking the time today to tune in from around the world to share this really valuable information. I mean, it's really great what SpliceBio is doing, and we want to just thank SpliceBio again for sponsoring today's session, and for partnering with the Carroll Center to really make sure we're bringing this meaningful information to our community. So we're very grateful to you both, and to your team for all the work you guys are doing. I want to take a moment to share our exit code for those that are interested in ACVREP credits. Our exit code is BLUE, BLUE, just like the color. So please make sure you email development@carroll.org before the end of day with the entrance and exit codes for today's session, and we can absolutely help. Like I mentioned, we'll be in touch with everyone who's registered very soon with all of this information as well as the recording. And we just, again, want to thank you both for tuning in.
Thanks very much, Michelle. And just to say, I'm sorry we couldn't get to all of the questions. There's some excellent questions here. So, but we'll endeavour to get some responses back in, over the next, I guess, few days, Michelle, right?
Absolutely.
So this week, next week, yeah.
Perfect. Absolutely. We'll make sure to note them all down and to get in touch with all of those whose questions we couldn't get to. So much good information. We'd be here all day probably.
No, that's great. It's great stuff. All right.
Great.
So thank you, Michelle, and thanks to the audience as well for their interest and for the great questions.
Absolutely. Thank you everyone for tuning in. And we look forward to hopefully our next session.
Thanks again.
you.
Bye-bye.
Bye.
Bye-bye, everyone.
Bye, everyone.